6:42 in the morning and I'm here in Queensland, Australia to track some bearded dragons.
Hey, my name is Kristoffer Wild. I'm a PhD student from the University of Canberra studying the ecological causes and consequences of sex reversal in bearded dragons. So these guys are a unique animal where they undergo genetic sex determination, almost like a human. So their offspring is already genetically determined when females go out in the field and lay eggs. What our research group recently found out is that these critters actually undergo sex reversal. And what I mean by that word sex reversal is when the mom goes out and lays the eggs out in the field, half are going to be male and half are going to be female. Well, sometimes temperature can override these processes. And this isn't what you think about when turtles lay eggs and temperature dictates their sex. The temperature will override our bearded dragons. So it's not what's going on in turtles or crocodiles. It's very different. So what happens, mom lays eggs, half of them are male, half of them are female. Then temperatures over 33 degrees Celsius will cause everything in that nest to be female. And for half of those individuals, half of them will have the genes that say they're male. And what we're finding are these sex reverse females that are reproductively valuable. They look like females. They have these phenotypes that help their fitness out, so they're stronger, they're bigger, they're more fecund, and what we're studying out in the field is the success of these sex reverse females.
First things first this morning is we're going to have a little drive around the roads and just see what we can spot from the car.
You miss her when it's not here.
We've just stopped the car because we've noticed the bearded dragon on top of a log right over there. We're gonna get him bagged up so we can get him back to the lab later.
Dude, for sure, yeah. The weird thing is, you know who just died over here? Like right there? That's where he's been.
He's nice looking.
Mm-hmm.
He just doesn't move. He dead? What? Oh my god. Shut up.
It's not him.
I know, but that's...
So we had a lizard with 3 legs that lived literally right over there.
Oh my god. Oh. Maybe there's a bird that particularly likes legs.
Just
around.
one leg.
Yeah. It was the other leg on Charmander though, wasn't it?
Yeah, it's back right. That's really funny. Hey, you're gonna replace Charmander. We're gonna study.
It opens a lot of naming opportunities. Oh. Now he realizes he's in the hands of a fucking predator.
Okay, cool. I need to just record some stuff.
There we go. So we managed to get a new dragon this morning, which is fantastic. And now we're off into the bush to track down lizard number 14, who's known as Munchkin. Let's go see if we can find him, eh? So the signal's just said that Munchkin is right over here by this tree. We're gonna see if we can spot him and then record his spatial location. So that's one lizard bagged this morning and one lizard tracked. What we're gonna do now is just go for a walk around the bush and see if we can find any more new lizards. Let's go. fantastic wildlife out there in the bush this morning. We got emu, we got the echidna, we got the major Mitchell cockatoo, but sadly there wasn't any dragons, but that's just how it goes. We're gonna head back to the lab now and get our boy from this morning all processed so we can get him back in the field as soon as possible.
So once we capture a new animal out in the field, what we'll do is note some of the habitat variables. We'll then bring the animal back to our field site. We then fit them with a little jacket. We program them with their unique GPS VHF, so their radio telemetry unit. And then we measure their morphometrics, so looking at body size and mass, so we can compare the males and sex reverse females and females. And then we release them within the next day. But their jackets are actually really unique. This is the first time we've done this with these lizards. So we custom make their jackets to fit their neck and bellies perfectly. And what we've found is that the attachment method hasn't hindered any of their movements or given them off to any predators. Because we go really into detail camouflage. And one other thing we do in the lab too that's really important is this sex test. So because we can look at a female and say, oh, that's a female, we don't know what the genes are. And that's what gives us the idea, or cues us for if that individual is sex reversed. So what we have to do then is take a small blood sample that we can then take back, or I can post back to our lab, and our other science team up in University of Canberra will genotype the animals and tell me what the chromosomes are. So if it were a sex reverse female, it would have the male chromosomes, but it would be phenotypic female. When I say phenotypic, it's a functioning, looks like a female. So that's the only way we can tell if the animal is sex reverse, is by sending that blood back to the lab to get then genotyped.
We've decided to call our boy from this morning Blackbeard, because he's only got one leg. He's all fitted with his jacket and his radio tracker. So now we're just going to go place him back where we found him this morning and leave him to his little lizard life.
There he is. I'm gonna put him down low.
So that's it. Blackbeard is back on his log. He's got his jacket. Kris and the guys here will track him in about a week just to figure out where he's moved to. And all in all, I think that was a pretty successful day in the bush. Can you give me a funny anecdote with your lizards in the bush, or just like a really cool bearded dragon fact?
Oh, anecdote. Okay. What I've figured out with this study, or working with these lizards out in the field, they're really popular in the pet trade, and you think, oh, that's a large lizard, and yes, I am American wandering around in the bush, but I figured, oh, I'm going to move here and catch heaps of them. Well, for an animal that's as big as maybe your arm, other volunteers can also attest to this as well. They're really tough to find, and they really rely on crypsis. So These animals don't move when we're hiking, and I found this just really interesting, that these animals, being as big as they are and as charismatic as they are, that they're pretty difficult to find in the field. I don't know.
Thank you.
Yeah.
That's